All right, we're going to skip now to Chapter 6 on genetic analysis and bacteria and phages. So we'll get back to Chapter 5 next, but Chapter 5 is a little bit more difficult of a chapter, so I wanted to spend a little bit less time on Chapter 6. So up until now, we've been mainly focusing on diploid organisms. So these include things like the pea plants that Mendel studied, Drosophila, mice, humans. But now we're going to look at what happens when you have haploid organisms like bacteria and phages. So by the end of this chapter, you should be able to explain how bacteria and phages are grown and phenotyped in the lab, identify and understand the different ways that genetic recombination can occur. So bacteria don't have sex in the same way that humans and diploid animals do, but they're still able to actually exchange genetic information and replace their DNA with other DNA. And then interpret the results of gene transfer experiments in bacteria and phages. So bacteria, so some of you might be familiar with bacteria already. If you've done any research, you've probably done some, especially if you've done molecular biology, you might have done a mini prep in order to isolate plasmids from E. coli bacteria. But there are tons and tons of different bacterial species, and bacteria are found pretty much everywhere that you can find any sort of life. And so these are 3 different examples of bacteria that you can find. So this is about 1 micron long, this is 5 micron longs, and then this actually is about 2 micron long here. So bacteria in general are very small. There's variability in their size, but typically they range between 1 and 5 microns long. They are haploid typically, and typically, unlike diploid animals, they have circular chromosomes. So their chromosomes actually are found in circles, and they typically possess only a single circular chromosome. And this is a general rule. There's some bacteria that have multiple chromosomes, but overall it's a good assumption that most bacteria have a single circular chromosome. The size of a chromosome varies, but 4 megabases is a semi-common length. So this bacteria, for example, right here is Caulobacter crescentus, and this has a 4 megabase genome. So bacteria chromosomes and genomes are actually much more condensed as well. So you think about humans, which have 3 gigabytes of DNA. These bacteria have only almost 1,000th of the DNA. So they have much more compact genomes. They have much less intergenic regions of their DNA, and they have much fewer genes. So typically only about 4,000 genes. They also can contain plasmids. So these plasmids are separate from the chromosome, and they're separate because, one, they're much smaller. So a plasmid might only be 50 kb. So part of the reason we distinguish plasmids from chromosomes is that they're smaller. Another reason to distinguish them is that they have multiple copies. So you can have up to 1,000 copies of the same plasmid within the cell. So there's these high copy plasmids that have extremely large number of copies. And it's also variable. So if you have cell division, one cell does not get the exact same amount of plasmids as the other cell. So there's a stochasticness that doesn't happen with chromosomes. So these are 2 reasons to distinguish plasmids from chromosomes, and then these are also typically not essential. So the bacteria needs a chromosome in order to survive, but they don't need a plasmid typically in order to survive. So DNA is organized in this way, and then bacteria also reproduce asexually. So they don't have to have sex with another member of their species. They don't, form gametes, and instead they reproduce asexually, producing a mostly identical daughter cell. So plasmids again might be different, but each cell will have the exact same chromosome. So this is an example of a Caulobacter. This is called Caulobacter crescentus. This is actually the bacteria that I studied in graduate school. And this was famous for its asymmetric cell division. So this right here was called the mother cell. And this mother cell had a stalk, so calo, kind of like cauliflower, has a stalked bacteria. And so this bacteria would use the stalk in order to grab onto a substrate of some sort. So this could be a pebble in a stream, for example. And then when this mother cell divided, this new cell that was being produced would have a flagellum, and this flagellum could be used for this bacteria to swim away. So this bacteria would be fixed, and then the daughter cell would be able to swim and colonize new locations. So in order to do this, the DNA had to replicate, and then you had to segregate that new DNA molecule into the new cell. This is an example of some bacteria that are found in the Dead Sea. So they have these crazy high salt concentrations. And so these bacteria actually have these almost cubical shapes, I think, in order to support the osmotic pressure that's created by the salt. Bacteria also can be infected by viruses known as bacteriophages. So we'll see kind of pictures of this, but you have these really small viral particles that can actually latch on to a bacterial cell, inject the viral RNA, and then take over the bacterial DNA replication system and transcription system in order to produce new viral particles and eventually killing and lysing the cell. And one final distinction is that they don't have a nucleus. So there is no nucleus. This DNA molecule is actually just within the cytoplasm of the bacterial cell. So that is a quick introduction to bacteria. And so this chapter will be separated into 4 parts. So we're going to talk about how do you grow and phenotype bacteria and phages. We're going to talk about conjugation and transformation. So this is a method of DNA exchange in bacteria, actually 2 methods. Transduction, a third method of DNA exchange that's mediated by phages. And then fourth, we'll talk about genetic exchanges in viruses.